What is the role of these particular restriction enzymes into the cloning process, right? So let's start. Restrictions in endonucleases, right? It is also known as restriction enzyme, right? So these are bacterial enzymes. These are nothing but bacterial enzyme that cut DNA molecule, right? Ultimately, what we are doing, we are cutting one DNA molecule from the parent cell or desired organisms, and that we are going to clone or insert or introduce into the host cell. That is the mechanism in the cloning, right? So there we are going to use this restriction enzyme. These are the bacterial enzymes that cut DNA molecule only at restriction sites, right? That's what it is called, a restriction enzyme, right? So here in DNA you can see there will be the restriction site, right? This red part I am indicating here as a restriction site. From here only the DNA it is going to cut, right? So that's what cut DNA molecule only at restriction site. Okay, so let's discuss the different types of the restriction endonucleases. The first one is based on the type of cut from where it is going to cut the DNA molecule. Accordingly, we have classified into two categories: cut with sticky end and cuts with the blunt end. So cut with the sticky end, when we talk about, you can see here this is the DNA. So how it is going to cut in this way, right? From G it is going, and again it is covering up to the G. When it comes to the blunt end, it will be straight like that. Okay, so 5 3 and 3 5 like that in DNA structure, ultra structure of DNA you can see. So this is called sticky end cut, and this is called blunt end cut. So both cutting it is possible into the cloning, where we are inserting the DNA molecule into the host cell, right? Coming to the second type, based on the organism from which they derived, right? From where we have received, because I told you in definition itself, these are the bacterial enzymes. So which kind of bacteria we have got this restriction enzyme for the cloning purpose? So EcoRI. So it is obtained from the E. coli bacteria. Coming to the second type, the BamHI, that is Bacillus amyloliquefaciens, right? So this is the bacteria from where we are getting this restriction enzyme. That's what, with the name of bacteria, it has named in this way. Coming to the third type of the restriction enzyme, protect bacteria from bacteriophage infection. So it is also called restricts viral replication, right? So these are the different types. Coming to the classes of restriction endonucleases, how we can two types it is there, type 1 and type 2. So coming to the type 1, what is happening here, cuts the DNA. Ultimately the role is cutting DNA molecule. So the type 1 it cut the DNA on both strands, but at a non-specific location, right? Restriction site it is a specific location, but type 1 it cut at non-specific location at varying distance from the particular sequence that is not recognized by the restriction enzyme. That is the type 1. Coming to the type 2, cuts both strands of DNA within the particular sequence recognized by the restriction enzyme, right? So at a specific location it is going to cut. So type 2 it is most frequently used. Type 1 it is not that much used into the cloning process, right? So restriction enzyme or restriction endonucleases, it is one of the important enzyme which is being used into the cloning process to cutting the DNA to provide the vector, so that vector can transfer that DNA into the host cell, and further replication can be taken place over there to produce the same types of organism for cloning purpose. Thank you.